So I made a little bit of progress on the project and I wanted to share it so that I can remember what I did, and also for anybody else who wants to play along. So I've been just to start with where you can start. I've been putting some stuff on Piazza about genome sources, and one of them is a link to one of the websites where they let you download the human genome. So if you follow that link from Piazza, you'll get here. And so there are two important things here that I sort of understand. One is, so this is just the human genome. And this is also a place where you can get like the chimp genome or whatever. So if you want to download the DNA sequences, then these are over here on the left. And there's an FTP site, and then in the post that I put on Piazza, I explain that you can download this Readme file at the very end, and it gives you an explanation of what these files are. And it's a little bit more convenient than the way the genome was presented in the other files that we've been working with, not least because it's already broken down by chromosome, so you can download the 22nd chromosome, which is 11 megabytes, instead of downloading 3 gigabytes plus before expanding it for the other file. The other file apparently is just everything and the kitchen sink all thrown in. So over here, there's something else. There are the gene annotation files, and I don't understand a lot of these things, but if you click here for GTF, GFF, BFF forever, you get these files. There's another README here that explains what these things are. I think the one that I got is just this one. Let's check in my thing and crap. Well, this is not the best place to do it. Sorry the screen is so busy. So what are these GTF files, GFF files? So come over here to the notebook that I've been working with, and so all of these are gene annotation files, and they're all sort of versions of the same thing. You can open all of them with, I believe, with this GFF utils package. Let me make this a little bigger. Okay, so the ones that I have in my current directory that I'm working with the one that I really want to focus on right now is the gene annotation file for the 22nd chromosome. So I'm just going to be working with that, and to be honest with you, it doesn't quite match up with what you see available for download here. And so let me see if I can figure out where I got it so you can get it too. So instead of this is it. So it's probably hard for you to see the difference here because everything is so small, but it says download GTF, and then there's a separate link for GFF3. So to get the file that I'm working with, you go here to GFF3, and then I'm working with chromosome 22. So it's this file, I believe, which is only 872 kilobytes. So it downloads like instantly. All right, so once you get this file, so what do you do? Let me just skip down a little bit and show you what kinds of things are here. So these are the sorts of things that are described in this file. You've got let's just look at the important ones: chromosomes, exons, genes. So that the thing that I am interested here is genes. So what I want to use this file for right now is just to get a list of the human genes on chromosome 22. That's what I'm going to try to do. So these are, as far as I can tell, people don't know exactly which ones the genes are. I mean, some of them are like pseudogenes or might be pseudogenes, and they've sort of been automatically detected, and it's a little bit messy at present. And hopefully Dr. Lentz can correct me if I'm wrong. about any of that. But so one thing I think we might want to do later is try to identify genes on our own. But at any rate, we need to be able to see what these genes are, according to Hoyle, what the experts on this have come up with. So the way you do that, first you have to build a dictionary, and I commented out that line here because it takes a long time to do. So where did I get this? I didn't just make this up. So I'm following along with this. Hopefully you can see that URL. What this is just the page explaining how to use GFF utils. So if you just Google GFF utils, you'll get a link to this tutorial, and this is the source of these commands that I'm trying out over here in my notebook. So this is the one that creates the database. And then I guess that once the database exists, it lets you do things a little bit faster, and you can do cool things like just get the features that are genes, which is what I've done right here. So I call my database Homo22, and then I get the features, which are genes. So this is, when you get it, it's something called a Python generator. So maybe I should say a little bit about what that is. So those of you that have had some programming before, you know that you can write a repeat loop to look through an array, right? Or just if you don't know what an array is, just think of it as a list of things. So this is like a list. So what's the difference between a list and a generator? The difference between a list and a generator is, with a generator, the items are not all stored in memory at once. So that's important in what we're doing because they're memory constraints. So if this were chromosome 1 or something much bigger, then there might not be room in memory to store all of the genes at the same time. But with a generator, you just get one gene at a time, and so you can iterate over it and not have a memory explosion. But in the case of chromosome 22, you actually can load them all in memory at once. And so when that's possible, you can just use Python to translate the generator to a list like this. And then you get to use all of the nice functions that Python has built in for working with lists. And one of them is just the length. So this tells you that there are, according to this annotation, 505 genes on chromosome 22. And so let's just look at one at random. So I decided to look at the one that has index 1, 2, 3. And so, we've talked about tab completion before. So one way to sort of play around with this thing is to hit period and then tab. And then you can look through here and see if there's anything that interests you. And if there is, you can type question mark. Let me just cover this up for the time being. And then the question mark will bring up the doc string. And then you can scroll through here. You can make this full screen if you want to. And you can read some documentation about that function or property of the gene and learn more about it. Okay. So I'm just dumping it to the screen in sort of raw form, and you can see that there are several things here. This one is a key that you can use to look up this gene directly from the database. And I do that down here. Let me skip ahead. So this line, what I've done is I've taken that first identifier there, and you can use this notation. This is Python dictionary notation. So you're looking up something in this database, and it's just the thing in the database that has this key, and then you can work directly with that gene. So that's what we're going to do down here. It's just the same gene, the one that happens to have index 1, 2, 3 in the list of genes that the database returned. So what are these other things? 22, Havana, etc. So those things are explained here. So the 20. So okay, I think we should start with source. So I think Havana is the source. Feature is what type of feature it is, so it's a gene. Then you have the start location, which is this. That's the nucleotide. That's the base pair or whatever at which it starts. And the next one is the end. And this one is a score. I'm not sure what that means. So this is whether this is going in the forward direction or the backward direction. So I'm a little shaky on this, but I have a totally stupid understanding of what's happening. But evidently, like DNA is a double helix, and so both sides of the helix contain essentially the same information because they're mirror images of each other. And one end of one strand is 5 feet long, and the other one is 3 feet long. And so you can talk about what direction you're going along the strand by specifying whether you're going from the 5 foot end to the 3 foot end, or from the 3 foot end to the 5 foot end. I know that's not literally chemically what's happening, but has something to do with it. Frame, this is not something that we've had to worry about yet, but there's a nice Wikipedia article on this. So with DNA, there are 6 possible reading frames because the base pairs are grouped into I guess amino acid sequences or whatever you want to call it, and they're just triples, right? They're broken up into chunks of 3. So where do you start counting 1, 2, 3? You can start at the 0th place, the first place, that means skip the first one, or you can start at the 2nd place. And then you can also specify which strand you're talking about. So that gives a total of 6 possible reading frames, but this is not something that I've had to worry about yet, and hopefully we won't. See, here it is talking about the 5 foot end. You thought I was joking. It's true. Okay, so we have the frame, and these are just weird ASCII characters here, but I think that's what they are. I don't know if that just means that those are not defined here or something. I don't know. Maybe I'm wrong. So the last thing is something. It's an attribute semicolon separated list of tag value pairs. So here are some attributes of this gene. It has ID, description, source, version, name, biotype. Okay, and then there's that very last number, and I don't know what that is. Where did I get all this crap? I got it from the Readme for the GTF dump, back on the first page that I showed you. Like right, where were we? Right here, sorry. If you look at this Readme, and reading these Readmes is annoying, because if you just click on them, they don't open. If you download them to a Windows machine, God knows what will happen. But what I'm doing is I download them, and then when I want to read them, I open them with a text editor like this. So gedit, for example. And then I get this, and then it explains how that file type works, and then it explains about fields, and the thing that I showed you I got just by copying and pasting that into the notebook file. There's a lot more information here. Okay, so this gene, let's look at this gene a little bit more just to practice working with genes. So here it is again. This time I got it directly from the database. And I'm going to look at its attributes again. So these are just the same attributes that we looked at before. So you can take this, the name, and Google it. So I did that. And when I did that, I got this page. And so this is Gene Cards, Human Gene Database, or whatever. So I kind of scanned down through here. It has some interesting, actually more incomprehensible than interesting information about this gene. So what I wanted to do is I wanted to get to the Genome Browser view. And this is something that I began to understand today. So a chromosome has this little pinchy part in the middle, which is where the microtubule grabs it during mitosis or meiosis or something, and it's called the centromere. And each chromosome has a short end and a long end. The short end is called P, the long end is called Q, and then there are bands that appear on the chromosome, evidently when you stain it using a certain technique, and there are gene coordinates that are, I guess, like pre-digital, that just specify the location of genes with respect to whether it's long or short, which chromosome it is, and which band and which subband. Anyway, so one interesting thing that I found on this page is what I think is the accession number. It says it's the ref-seq DNA sequence for blah, blah, blah. So if you click on this, then it takes you here. And there's some more information about the gene. But the thing that I liked that I found here is there's a way to go right to a view of the same gene in the Genome Browser. So let's go to the graphics here. Okay, so there's a lot of distracting crap which you can sort of close. Like, I'm not interested in this. What I basically just wanted to see was the sequence of the thing. And you can see that they're up here. I hope you can see this number. Let me try to make it a little bit bigger. So these numbers here are like the base pair index. So look at this. This is 22,792,600. So what does that have to do with anything? So if you are a freak, then you remember that this number was 22,792,491. So that is the base pair start location of this gene. And let's come back here. So I could scroll over. Can I pull? No, I have to do it kind of manually. Go away. I messed that one up. This is the same thing. Did I ever get some picture that I like better? Sorry for jumping around. Anyway, so I fooled around with this until I got to the start of the gene, and I wanted to just see the base pairs that the Genome Browser would show me. And when I did that, I took a screenshot of it, and here it is. So you can look, here's 22,792,000. Let me see if I can make this bigger. No, sorry, I don't seem to be able to enlarge that. Maybe if I do F11, I don't know. So here's 22,792,490, and remember it starts at 491. So depending on which strand see, here's the 5-foot end, and here's the it's reversed for the other. I need to figure out what the technical word for this is. But this top one goes from 5 to 3, and the bottom one goes from 3 to 5. Anyway, so if you look at the 5 to 3 strand, then the gene begins T, G, C, T. All right, so if that's true, can we connect that with the files that we have that actually have the DNA sequence? So those are in the so-called FASTA files. So what I wanted to do was to be able to go from a gene as specified in one of these annotation files to the actual sequence. The sequence is not given in the annotation file. It's not given in the GFF file, but what is given are these base pair start and stop numbers. And also the exons and other nice things, but they don't tell you the actual sequence. So we need to be able to go from this description to an actual sequence description. Why? Well, for example, maybe we want to find some human gene and then find something similar in like the chimpanzee genome. So then this gives us a list of human genes, and so we can take one and go try to find what are they called? Homolog. We could go try to find a homolog in the chimpanzee genome. So anyway, so how do you get from this kind of a description to the actual sequence? So I have this file, the FASTA file, and let me remind you where FASTA files live. FASTA files live over here. If you click on FASTA, and you just download chromosome 22, and it's 11 megabytes in compressed form. Very easy to work with. So that's the file that I'm opening here. How do I know how to open it like that? That's because I just Googled Biopython open FASTA file, and then I followed the tutorial directions for doing that. I think I've closed now. So I've got chromosome 22. And so this would return a generator, the stuff that I just highlighted. But in this generator, there's only one thing, which is the chromosome 22. So what this next function does is it gets the next thing in a generator. So you know generators vomit things up one at a time. So this just gets the next one, which is the first one, which is the only one, which is chromosome 22. And so now I have a handle on that. And I can do chromosome 22.sequence, and then I what I'm doing here is called Python array slicing. So if you want to know more about Python array slicing, you can Google that. Let me do something more simple. Than I don't want to get too off topic, but let me just show you something about array slicing, okay? Okay, so I have a list of letters here, and the letters are the letters that occur in the word Methuselah. So you can index things in a Python list, and counting starts at 0. So this is 0, 1, 2. So if I look at the second entry in this list, or the thing with index 2, which is actually the third thing, it's T. Okay. Now, what if I want to look at a range of things? So this would be, let's do 6 here. Okay, so what the syntax for doing that is to do the start and then a colon and then the stop. And so what this is everything with index between 2 and 6, but not including 6. So it includes the start on the left, but the stop on the right is not included, and that's very typical of Python. Okay, so we go 0, 1, 2 is T, 3 is H, 4 is U, 5 is S, 6 doesn't get to come. So that's what's happening here, and so the sequence is it supports the same array slicing notation. So I look at the start location of the gene, and so why did I subtract 1 here? It's because biologists did a silly thing with their conventions, which is that they started at 1 instead of 0. This number, if we were to go down to the very end here, it would begin with 1 and not with a 0. And so that means that if you're going to convert, so this is in sort of 1-based understanding, and I'm using Python, so I have to get down to the 0-based understanding, so I take 1 away. And so the gene start, according to the biological conventions, -1 is the start index in the sequence of the gene. So let's just do a sanity check here. I've only looked at the first 10 base pairs, but here they are. T, G, C, T, G, 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 T, C. So look here. Here's 491, right? 22,792,491 begins T, G, C, T, G, 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 T, C. So that's it. So now we have a way of connecting genes, we have a way of listing all genes, or all known genes, according to Hoyle or the authorities that be or whatever. And now we can easily write computer code to get the associated sequence. And that's all I have to say about that.